Good afternoon. So this is our second seminar in a series of many to come. So today, Dr. Justin Patucci will be talking about studies using clinical data. Last week, we had an overview of data science and study design. So without further ado, Justin.
Thanks, Dr. Honavar. So thanks everyone for joining. My name is Justin Patucci, and I will be discussing a quick overview of some of the types of clinical data and sources of clinical data we have access to here at Penn State, and also touching on some various aspects of studies, introducing us to the OHDSI uh, study network, and diving into some examples that we're working on within the CTSI Informatics core. So a quick sort of motivation, as many of you know, they're across different fields and domains. There's a situation where vast amounts of data exist, and that data really has the potential for useful and potentially transformative, to be transformative with the wealth of information it contains. But and healthcare really is no different, but there's certainly a list of challenges. And within sort of the clinical research questions on the data side, some of these challenges include just getting access to data of sufficient quality and quantity. Once you have access to the data, right, you need to be able to sort of wrangle that down and harmonize it and potentially integrate it with disparate other sources of data. So having that expertise in-house within your research group can be a challenge. Having sort of methodological expertise, whether that's statistical machine learning methods you want to apply, having that expertise can be a challenge. Sometimes there's computational sort of resource challenges. If you have a massive sort of, maybe let's say like an image data set, where do you house that data? Do you have sort of sufficient so storage needs? Do you have GPUs or potential accelerators to train a model if that's what you're doing? Do you have the expertise to do that? Of course, there's other challenges around sort of ethical and legal challenges anytime you sort of deal with healthcare data. Generalizability of results. A lot of time, sort of a paper will come out and it will focus on a patient population that is sort of very narrow, maybe exists in one hospital system in some corner of the country. How do those results generally apply to sort of the broader population? Along with that, there's challenges across reproducibility. If we can't share sort of data or potentially code, how do we sort of reproduce results and things like that? So within hopefully I'm aiming to touch on some of these challenges and how within the CTSI Informatics core we're aiming to address some of these challenges. So goals for today, as I mentioned, we're going to look at different EHR sources we have available. We're going to become familiar with OHDSI, what is OHDSI, and what is a multi-site or network study within the OHDSI collaborative. We'll see some example EHR studies that have taken place within OHDSI and we're conducting within the CTSI Informatics core. And then just next steps, how to move forward from there. So jumping right into EHR data options. So Penn State Health now has their own OMOP Common Data Model database. So we'll get into what those acronyms mean later. They're sort of tied up within the OHDSI collaborative, but essentially we have a database with 1.6 million patients. I checked earlier this morning. That's the unique patient ID, person ID count. And really this is, as of right now, tabular data. So this is your sort of that historical record, patient record that captures diagnosis codes, procedure codes, vital sign measurements, medications that were prescribed, those sorts of things. And this data exists in this OMOP Common Data Model format. It's essentially a SQL database. Backends can vary, but you can interact with this through SQL queries, and we'll see through the OHDSI collaborative, there's also some sort of high-level tools that make interaction with that database a little bit easier. So that's one option in terms of data that's immediately available to clinician researchers. But many times we see that maybe our data, the Penn State Health data, is inadequate for some reason, and many different reasons. Uh, the most common would be just not enough patients for the clinical question at hand. So where do we go from there? Um, sort of this is enters into the Odyssey Health Data Sciences and Informatics Collaborative, or known as Odyssey. So this is a global multidisciplinary collaborative. You can see sort of stakeholders across academia, government, and industry, really generally focused on bringing out the value of healthcare data through evidence generation. And this collaborative does that through methodological research and development, open source software and tools, and also through their common data model, which is a consistent way to structure electronic health record data, which we'll see a little bit later, some more details of that in the presentation. So sort of a high level, how global is Odyssey? And I apologize, this slide, or the numbers on this slide, is about a year old at this point. So these numbers have all increased. You can see sort of the claim is 12% of the world population is captured in the combination of OMOP databases that exist at these different sites. So it's quite extensive. We'll talk about network studies, and really network studies is the holy grail of a network study would be to have complete participation by every site that has an OMOP common data model formatted database. That would be sort of the extreme end of capturing 12% of the world population. In reality, I haven't seen a study that big, but the collaborative is very ambitious, and there are quite large network studies that have been conducted and completed. So at a high level, the Odyssey model is the following. Each site formats their data according to their standard, the OMOP CDM standard. And then a analysis protocol can be developed at any one of those sites, and that analysis protocol leverages the open source packages that the Odyssey Collaborative also develops. So we have everyone's data is formatted the same. Everyone's sort of using this common analysis software. So we can get into this model where this protocol is developed, and we can share analysis code with the assumption that everyone's data is formatted the same. So at a high level, that's how network studies work within the Odyssey Collaborative, and we'll jump into some of the details of that again later on. This is sort of just a high level, what is Odyssey? So Odyssey is another place where we can get data, more data than what Penn State Health has to offer. Another option is TrinetX. So TrinetX, through TrinetX, you can get access to, again, this is more tabular health data. So very similar. These are your diagnosis codes, your procedure codes, vital signs, basic demographics, age, gender, race, ethnicity. And through TrinetX, there's a web platform, and that web platform has a basic cohort builder where you can start to qualify patients for different conditions, outcomes, and there's some basic analysis tools through that platform. And there's a couple different databases available through TrinetX. The primary one that we use is the Research Network. And again, this slide is a few months old. So at the time, there were 71 healthcare organizations contributing data to that network, and in excess of 100 million patients. The Diamond Network, this was claims data. It's since been depreciated, but the informatics core does have a random sample from that Diamond Network. So potentially we could leverage that random sample, that subset of this 213 million patients that are available in that Diamond Network. I should mention that, so by immediately using the TrinetX platform, you sort of get you get data for a multi-site study. But when that data is delivered to to us or you. It's in sort of their proprietary flat CSV file format. And at the patient level, we can't tell which healthcare provider that data came from. So at a high level, I think we have north, south, east, west in terms of location identifiers. So there's not much that we can do with that location information. And this has to do with preventing the re-identification of that patient data that's provided from TrinetX. So other data sources or components, modes of data that we have experience with in previous projects or some ongoing projects, NCDB and SEER, and also, of course, in different domains, there's specifically curated research databases such as vascular surgery databases that some of members of our group have been working on. Signal data with ECG data, we'll see later in one of the pilot projects that we're working on. That group is looking to stand up essentially a database of all the ECG scans that have been performed to date, and this will be a general resource that's available to anyone within the Penn State research population to pull and utilize that ECG data. We have several projects that are using image data, whether those are primarily they've been CT scans, and what's very attractive is to not just use that image data, but to link that image data with the other existing electronic health record data, and that's done through the unique patient identifier, either on the identified side or ultimately through the on the de-identified side. Another big push in the community is to integrate social determinants of health data, and that's an ongoing effort that we're very interested in, and linking that again to existing electronic health record data. So focusing in here on these OHDSI network studies. So again, I mentioned this OMOP common data model a few times. Again, what is it? It's a standard way to represent. It's a standard data structure for electronic health record data, and the OHDSI collaborative sort of quite exhaustively curates and maintains a vocabulary. So different sites that use different coding systems can all sort of standardize on one sort of language or vocabulary. They have built-in tools and methods, what they call their Hades series of packages that again have these canned analysis protocols that deal with characterization studies or patient level prediction studies or effect estimation type studies. And at a high level, the aim is to standardize on the data structure, standardize on the analysis tools. So it makes sharing and collaborating much easier in this space where there's obviously a lot of concerns around data privacy and patient protections and things along those lines. So it's sort of an unwritten rule. Anytime you talk about OHDSI in the OMOP common data model, you have to sort of show a high level visualization of the database. So here it is. I've sort of hit that unmentioned requirement. But at a high level, you can see that most of these tables are linked through the person ID. There's different domains, and these domains are such as like procedure occurrence table, drug exposure table, condition table. And this is, again, that high level view of the OMOP common data model. It's something that's designed to be extensible. So there are efforts to incorporate the earlier versions of the OMOP common data, for example, model did not have clinician notes, but now you can see that's part of the standardized, the standard model here. And there are efforts in the community to incorporate other data, such as image data and things along those lines. So if we look at this slide, the first row on the left here, and we're focused in on data partner one. So we saw that map of the OHDSI network, and each dot on that map was a data network partner. So someone a healthcare organization, a private entity, has their healthcare data formatted according to the OMOP Common Data Model standard. So they took their source data and their, which has whatever coding system or structure it has, and there was some ETL process that converted it to the OMOP Common Data Model. So that this sort of central cylinder in the top row. A protocol is developed based on some clinical question, and the standard analytic tools, the OHDSI tools, which is primarily the Hades package, is applied to that common data model. And we can see that other data network partners that have already transformed their source data into the OMOP common data model can take that analysis protocol, run it against their OMOP common data model, and share these aggregate results. So this is sort of the model for how these network studies are conducted. So essentially, in this example, I think we're showing one exchange of results. So each site pulls down the analysis protocol, runs it, and shares the results. So this gets around, again, sharing the actual patient data. You're just sharing the results of the analysis protocol. There are efforts underway to perform network studies such that there's more than one sort of share point or sharing of data, and this allows for the training of models beyond a single site. So for example, we'll see in a few slides an example of a patient level prediction problem. So with this model that I'm showing here, each site would essentially fit their own patient level prediction model on their own data, and then that each site would essentially share the results, the coefficients of their model to the data network or to the site leading the study. There are efforts underway and standards methodological approaches being developed within OHDSI to across these sites train one model using everyone's data. So depending on the underlying algorithm, there's so many sort of shares of data, aggregate data that needs to occur for that one model to be trained sufficiently. So those federated learning, that sort of concept is something that the OHDSI Collaborative is focused on and working on. So this slide comes directly from the recent OHDSI symposium in last month in October. And at a high level, it sort of overviews what needs to be done in order to lead a network study. So the first thing that needs to be done is to clone the OHDSI GitHub repo. So there's a template repo that anyone looking to develop a study, they can clone that as the starting point. So what will that do for you? That'll give you sort of the templated breakdown for it will give you a templated Git repo that contains everything that you need sort of in a sort of plug and play type way. It will officially sort of register you in the OHDSI Collaborative, because you'll sort of be right in underneath the OHDSI Studies GitHub organization. The next thing you can do is develop a study protocol. And again, there's a template for this, and you can officially register that as a OHDSI Collaborative by cloning and pushing back to one of the OHDSI organizations. As part of that template study, you'll build out customizations for your own study. That's this third point. Once your protocol is established, you can reach back to the data network and start to see what data sites potentially have patients that are relevant to your study. So whenever you're recruiting sites that potentially you want to include in your study, you can reach out to the data network partner with maybe a cohort some cohort requirements, and they can check against the data network partners who has a relevant patient population to your study. So that's one way you can get study participants. Another way would be to announce that through the community calls, and also pushing out an announcement on the OHDSI forums. So again, this page is a great resource for anyone to come back to for those steps to conduct a network study. Um, yeah. So now I'm going to jump into one of those network studies that already exists within the Odyssey network. And this is sort of just give you a high level view from clinical question to a what does this look like within the Odyssey collaborative. So this is a this is a patient level prediction problem. So this is formulated as follows. Among a target population T, we aim to predict which patients at a defined moment of time will experience some outcome during some time at risk. So prediction is only done using information about the patients before that index date, right? We can't use future information to predict about the future. That's cheating. So the clinical question here was amongst women aged 40 to 74 who undergo a screening mammogram and who do not have a prior history of breast cancer, which will go on to develop breast cancer 90 days to 3 years following that screening. So coming back to that basic patient level prediction uh, format or framework, we can see sort of what that looks like with this clinical question plugged in. So our index date is their first mammogram in 2 years. We're requiring no breast cancer or no prior breast cancer or at least 90 days after that scan to be not have an instance of breast cancer diagnosis. The time at risk is that 90 days to 3 years. And we're requiring, in this study, they required 3 years of medical history prior to that mammogram screening. So defining the study details. So if was your clinical question, you have to start to think about how do you qualify patients for the index event? So how do you qualify patients for mammogram screening? How do you qualify for that outcome event of breast cancer? So what covariates or features are you interested in using to train a predictive model? What models should be used? What sort of hyperparameters should be selected, etc. So these details and more are captured in the Odyssey study protocol. So again, there's a template in the Odyssey GitHub organization where you can directly pull down and start to fill out the details of a study protocol. And that's really, again, that first step that needs to be done within the Odyssey network study before you can start recruiting for a for network participation. So I'll leave since sort of I want to be cognizant of time, we'll leave the details of the results of this study for you, but here's the links here. You can see what a analysis protocol looks like for this study. There's the GitHub repo. So the second link here, and I'll takes you to the GitHub repo, which has that analysis protocol using the Odyssey open source packages Hades. And then the results are shared here in this Our Shiny app. Okay. So now focusing on just some of the different projects that we have going on here within CTSI Informatics Core. So these were our CTSI Seed Grant recipients, and there's 4 active projects here. So Dr. Lanham, we have the analysis and comparison of anticoagulation with and without bridging therapy after shoulder surgery. So this is primarily using tabular healthcare data and to again identify risks of complications with and without that bridge therapy. We have a 2 projects that are aiming to utilize image data. So this is Chris Zhang and Dr. Goyle's project, and also Dr. Manali Vaskar's project. So with Chris Zhang and Dr. Goyle's project, this is a machine learning analysis of incidental thyroid mass CT imaging, and it really aims to minimize the need for ultrasound and increase the ability to console for fine needle aspiration for incidental threshold nodules by training machine learning algorithms to classify masses as benign or suspicious for malignancy. And for Dr. Vasikar's, we're aiming to develop a deep learning algorithm based on predictive model for morbidity and systematic cancer therapy-induced pneumonitis. So for Dr. Vasikar's study, we are using electronic health record data, so that tabular data, and we're combining that with image data. So this is an instance where we're using multiple modes of data to hopefully develop a more robust and more predictive model. Some of the projects that we have going on, we have a project with Dr. Seth Pentinelli, and this is about developing a model to predict whether a pupil expansion device is required in cataract surgery patients. There's some ongoing projects with, again, Dr. Vasikar's group, centered around and focused around racial disparities in the treatment and outcomes of breast cancer patients. We have another study looking at treatment disparities with device implantation for epilepsy patients. And finally, there's some ongoing research with Dr. Aziz centered around risk and outcome prediction for patients with peripheral artery disease. So we saw some example studies that CTSI Informatics Core are currently focused on. We're really aiming to help across the sort of gambit of the sort of typical data science or machine learning project lifecycle, from getting access to that data, defining that patient cohort, to hands-on implementation, data cleaning, feature engineering, etc., training a model, training a machine learning model, or another application of some methodology to answer an outstanding clinical question, and then sort of all the way down to interpreting and potentially utilizing that model. So we saw some of the projects that we're working on. Again, really aiming to assist across that data science sort of project lifecycle. How do you directly engage? So the first step would be develop that clear clinical question, and we see that question that we focused in on that example Odyssey network study. It would be great if you come with a well-defined clinical question. If you come with an idea of how you can qualify patients for your particular cohort of interest or outcome of interest. I have some links here to some tools within the Odyssey Collaborative that could help you do that. So there's a phenotype library that is actively being developed all the time to have standard ways to qualify patients for different conditions, outcomes, and also this link to Athena. So Athena is sort of a graphical view insight into the OMOP or the Odyssey vocabulary. So you can directly type in strings and see sort of what standard concepts show up in the Odyssey OMOP vocabulary, and start to think about how you can qualify patients based on those standard concepts. And then from there, it's really just mapping those and plugging those into some of the packages available through Hades. Once we know how to qualify patients and sort of different timelines and thresholds that are relevant. Another thing that would be great before engaging would be to think about the different features that you think should be used. If this is for example, a prediction level problem, which diagnoses or procedures, etc., are relevant for that clinical question. So we are developing an intake form, but it's still in progress. So in the interim, we recommend reaching out to Avnish, and I have his email here. And I think that's it in terms of time. So thanks again. I'll be available for questions afterwards, but yeah, please engage, please email us, and thanks for your time.
Thank you, Justin. We'll have time after the meeting for questions and answers. And again, we will be posting this presentation on YouTube, so we'll send that link. And next is Prabhani. Prabhani, all yours. Thank you. You're muted right now.
All right, sorry. Can someone confirm that they can see my screen?
Yeah, we do see it.
Perfect, thank you. All right, hello everyone, and thank you for joining today's session. So before we start, let me briefly introduce myself. My name is Prabhani, and I joined the Penn State Statistics Department as a faculty member in 2018. And then shortly after, I got the opportunity to actually join the BERD Core. And since then, I've had the pleasure of talking to many researchers here at the UP campus, as well as some from the Hershey campus. And some of these discussions has led to long-term collaborations with multiple publications, grant proposals, and even student mentorships. So as a reminder, today's this talk is a part of a talk series that BERD and Informatics course are planning together to as a sort of like initiating a framework for initiating your clinical research. So for today's talk, I want to make it clear that my goal is not to teach you how to do a statistical analysis. In fact, I don't plan on talking about any statistical methods. Rather, what I want to share are some concepts and terminology that you will come across throughout different stages of your research and will need to think about. So some of the topics that you hear, some of the things that I talk about today might seem very basic, and some ideas perhaps are repetitive from what Justin mentioned and also from the last session. But I think those are important and often ignored, that they're worth revisiting or repeating. All right, so let's start by looking at the overall process. So when we are doing research, we want to learn something, some aspect about a group of subjects. So, but we often don't have access to the entire population of interest, or all the subjects in the population of interest. So what we do is we take a representative sample and collect data on this sample. There are different ways to take a representative sample, and Alan might have discussed some of these techniques in the last session. Once you collect the data, then we look at the patterns in the data to make conclusions. But we are not actually interested in making conclusions about our sample. So we want to extend these conclusions to the population of interest. And this is where statistical inference and statistical methods will help you. Of course, our ability to make inference is shaped by how we collected the data and our design. I'm sure in the last session, you heard a lot about the design and how important it is to take design into account in your analysis, and practical limitations that you have, things like that. But the point that I will reiterate is talk to a statistician at the very early stage of when you are planning your research. And you have the access to the BERD core as well as to the Informatics core. And at the BERD core, we work with a variety of projects. Some can be short-term, some can be long-term. So it's always better to actually initiate the discussion at the very beginning of when you're planning the research project. So in addition to the design, it's also important to think about the structure of your data, the pre-analysis steps, like whether you need to do some pre-processing, cleaning, what types of variables you have, what tools can you use to just take a first shot at your data. And my plan is to hopefully give you some ideas along these lines of topics. Okay, yeah. All right. So let's take this quick example to talk about introduce the idea of data structures. So this is a very simple example. It shows the number of active service members by their pay grade, the marital status, and the biological gender. And if you take this report, as it is for statistical analysis, we need to sort of reformat this data to be able to use in an analysis. The first thing would be to actually figure out how many variables we have here. If you look at this, we have, let's say that we can either think of it as a pay grade is a variable as it is, or we can think of it as the rank or the category and the level. So it's two variables, and then we have the marital status, and then we have the biological gender. So we have 4 variables. Now, think about putting each of these variables in one column and a subject in one row. This will sort of give us a format that we see here on the right-hand side, which is basically what we refer to as model-ready data. And in this new formatted data, you see sort of like a clean-looking format. And of course, the format that you want the data might depend on the question that you're trying to answer. Also, it might depend on the software tool that you might be using. So if you are interested in learning more about different formats that are available, you can actually take the article that I have cited here is a short article, excuse me, that explains some of these. So I would highly recommend taking a look at it. So so that's just one example. And in some projects, you might have more complicated data formats. For example, if you're using fMRI data or even other image data, text data, these different types of data needs to be pre-processed and before being able to do any types of analysis. And there are standard protocols that one can use in such situations. So once you have correctly structured data, before jumping into any statistical modeling, you should understand the patterns in the data, basically whether there are any duplicates, any inconsistencies in your data, whether there are any missing values, is there a particular pattern to missing values, or are there any unusual points? So to get an idea about these different types, a good starting point would be to actually create some numerical and graphical summaries to describe your data set. And of course, what types of summaries that you can use would depend on what type of variables you have. So variables can be quantitative and qualitative. Depending on whether it's qualitative or quantitative, the amount of information that each variable carry will be different. So qualitative variables can be binary variables like biological gender or a disease status, whether there is disease or not, no, yes, no type of things. It could be nominal with more than 2 categories. For example, like a blood type, where you have more than 2 categories. And it also sometimes could be ordinal, just like income level, pain level, means that you have more than 2 categories, but now you have also a natural order to the level of these levels. So depending on the type, you have different level of information. Then variables also can be quantitative, right? It can be continuous like BMI, weight, or the blood pressure level, the value itself, or it can be discrete like the number of pregnancies or the number of members in a household. So based on these different variable types, whether it's numerical or categorical, the numerical and graphical summaries that you can use will be different. For example, to summarize your quantitative data, we can use numbers like mean, median, standard deviation, or you can use graphs like box plots or histograms. So this will tell you, like this will give you an idea about the shape of your variables or the the shape of the data, right? And if you have quantitative variables, then you can use numbers like proportions, frequencies, or graphs like bar charts. So I do have to mention that the color scheme is actually, when it comes to visualizations, is important. And this is not something that I've addressed here. I think data visualization is another separate area probably that we can address in one of these talks. So these are some of the summaries that you can use just to get a quick idea about how your data looks like. And if you have, let's say, two variables and you're interested in associations between two variables, let's say that you have two numeric variables, then a scatter plot might be a good way to actually look at the association between the two quantitative variables. And if you look at the number that you can use is the correlation. But you have to keep in mind when you're using like a correlation as a number to talk about the relationship between two variables, it actually, the standard correlation, standard Pearson correlation that we look at is only focusing on the linear correlation. So in other words, you might have a higher value for your linear correlation, but this high value could be an artifact of one single point. Or you could say, when you calculate the correlation value, you could observe a very low correlation, but then it could be just because it's not a linear relationship. The two variables are still associated, but in a nonlinear way. So you have to, the point that I'm trying to make is that it's always better to plot the data, which will give you a really good idea. If you can, you should plot the data. And sometimes if you have very large number of records, your plots might not make sense. So in that sense, you might be able to take random samples of the data and then plot and see whether you capture these patterns. So, but in general, I think it's a good practice to actually plot your data and see what type of patterns that you see, because it helps you capture what problems you have in your data, whether you can actually use the data that you have to answer the research question that you're trying to answer. So these are some quick tools that you can use. In the same way that you can use the scatter plot, if you have two quantitative variables, you can also use, like if let's say that one of your variables was categorical or qualitative and the other was quantitative, then you can use side-by-side box plots. If both variables are qualitative, then you can use two-way tables with the frequencies or the proportions in them. So all of these will give you sort of like small snapshots of your data so that you can start understanding the patterns that exist in your data, which is a good starting point before you actually do any statistical analysis. I think I have about 5 more minutes. So I don't want to, as I said at the beginning of the session, that I don't plan on going on to details of the statistical analysis. And for you, I think you have access to the BERD core, and you also have access to the Informatics core that you can reach out and discuss about the statistical analysis. But what I want to talk about is that when you are looking at the statistical analysis, there are several things that you have to take into account. And some of the things I have listed here. For example, you might want to consider, like, you have a research question, a specific aim, and once you take that aim, you want to ask the questions, okay, to answer the question that I'm actually asking, is there like an outcome variable of interest? Is it just one outcome variable of interest, or are there multiple outcome variable of interest? Is there a latent variable that I'm trying to find here? What is the type of the outcome variable? Is it qualitative or is it quantitative? And then what are my candidate predictors? Are they categorical or are they numerical? And am I interested in actually making an inference? Or am I just interested in, is my primary interest is in doing predictions? So these are some of the questions that you have to ask when you are actually trying to figure out a good statistical approach. There are many ways that one can use to actually answer one statistical question, right? So it's as long as, like, there's a famous saying that not like none of the models are perfect, but all models tells you something, right? So it's the code is different. I'm just paraphrasing it. So so you will, like, when you have models, there are multiple ways to actually build a model. So you just, and like, one might not be necessarily better than the other one. So it's always a process. And in addition to these things, obviously, as you heard in the last session, you have to take the design into account. And when you are taking the designs, you might actually have to think about the limitations. And just as Justin mentioned, once you do the statistical analysis, when you are actually making conclusions, you have to take into account of the limitations to actually talk about the scope of the results or the generalizability of your results, and what numbers, what graphs, what results that you can use to actually present your method in publications. All these are things that the BERD core can help you with. So just to give a reference, I guess, if you go to the CTSI website and go to biostatistics, there is a consultation request where you can submit the consultation request. And one of the BERD core members, there are few of us. I should have the names listed here, but one of us will reach out to you and we can initiate the discussion. So I would say just use the resource that you have and reach out to us. Thank you.
Thank you, Prabhani. Hi, everyone. So we do have 10-12 minutes for question answers. Please feel free to unmute yourself and ask the question. You may use the chat button also. We are watching that. So we'll stay here for another few minutes just to see if we have questions. Again, as mentioned, these are our series of talks. We will reconvene. Actually, we'll have more talks, which we will be sending information by our newsletter, but they'll begin in February 2025. These are going to go on YouTube. You can watch them again. But yeah, since we have 4 experts here, 3, please use this time if you have any questions.
We're going to be YouTube famous with all these views. It's going to go viral. And
that's the plan.
Yep.
I think yeah. And again, these questions or any discussions which we are going to do here, they won't go on YouTube. Only the presentation by Prabhani and Justin will go on YouTube. So yeah, please don't feel that you'll be on YouTube. I think we can stay for a minute or so. Looks like not many questions here. Don't know why everybody's so quiet.
You need coffee.
Yeah, 3 o'clock. I think that's good enough. We will edit these, share on YouTube, and then reconvene again in February. Thank you, everybody.
Okay, sounds good.
Right.
Thanks.
Thank you.